Hi, my name is Emma Corbett, and my presentation is focused on the biotechnological control of flowering time. And for this presentation, we're going to specifically be looking at the molecular identification of the florigen signaling pathways and the applications of this pathway with CRISPR-Cas9 for crop improvement in alfalfa. So, for a brief outline, I will first establish the agricultural context and foundation for the florigen signaling pathway. Then, I will talk about the biotechnology used to manipulate this pathway and the subsequent integration of CRISPR-Cas9 technology. The core of my presentation will include a critical analysis of the 2023 study by Ulabu et al., which is my primary research paper, where I will detail their hypothesis, methodology, and significant improvements that they achieved within alfalfa. I will then conclude with the broader implications of this work and potential research directions. To start, in the wild, Plants to start, in wild plants, the transition to flowering is a highly regulated survival mechanism. However, in modern agriculture, natural flowering often happens in conflict with production goals. A key example of this is the perennial forage crop Medicago sativa, which is commonly known as alfalfa. Once the flowering process begins, alfalfa initiates what is called lignification of stem tissues, which is where the plant reallocates nutrients, primarily nitrogen, to reproductive structures. This process significantly reduces the digestibility and nutritional value of alfalfa, and this creates a big trade off for farmers and producers between having a high quality with low quantity of product or having a high quantity with very poor quality of alfalfa. With this, the molecular basis for floral transition was discovered through research in Arabidopsis thaliana, and this research identified that flowering locust tea is the central integrator of endogenous and environmental cues. They also found that the FT protein acts as a long-distance florigen symbol, with FT being synthesized in the leaf tissue in response to inductive conditions. Here, the FT moves via the phloem to the shoot apical meristem, where it is able to initiate genetic changes that are necessary for the induction of floral development. With this, FT has a conserved role across all angiosperms, making it a great target for genetic manipulation in all flowering plants. With this, research was done to manipulate flowering time to further confirm the role of flowering locust tea. Here I have a brief comparison of overexpression and gene silencing. Constitutive overexpression using strong promoters resulted in earlier flowering, but was often coupled with stunted growth and reduced vigor. With this, it was found that permanently silencing using RNA interference was able to delay flowering, but also frequently introduced pleiotropic effects that were incompatible with seed production. So these plants were able to not procreate. These approaches are proof of concept, but they did lack the control and precision that are required for a large scale application of modifying flowering time. Continuing on, the need for precision and control was further refined with the introduction of CRISPR-Cas9 genome editing. CRISPR works by facilitating the creation of targeted double-strand breaks in the genome, which are guided by customized RNA sequences. With this, the cell will repair these breaks, which often results in frameshift mutations that effectively knock out the targeted gene. A significant advantage of this is the ability to create non-transgenic plants with edited DNA. And this brings us to our primary research article, which represents the aforementioned applications. Here I will be looking at the 2023 study by Wulabu, which was published in the Plant Biotechnology Journal and directly addresses the economic challenge in alfalfa through targeted genome editing. For this study, the authors hypothesized that knockout of the MSFTA1 primary florigen gene in alfalfa would decouple flowering from environmental triggers, which would hopefully extend the vegetative phase and further delay flowering. With this, researchers also anticipated that this would lead to increased biomass accumulation and a sustained forage quality. However, a significant challenge with alfalfa is that it is a tetraploid plant, meaning that researchers had to disrupt all four copies of the FTA1 locus to achieve a strong and consistent phenotype. So, in order to address alfalfa having four copies of the MSFTA1 gene, the researchers employed a multiplex CRISPR-Cas9 system where they were able to design four distinct guide RNAs to target different exons of the MSFTA1 gene. This strategy was able to maximize the probability of inducing mutations across all four alleles. And with this, transgenic lines were rigorously genotyped to identify individuals that were carrying either biallelic or tetraallelic knockout mutations, and this was to ensure a complete loss of function. On this slide, I have also provided a diagram that was provided within the research article. Moving on, here I am showing another graph from the article showing the phenotypic data with all 6 selected homozygous mutant lines. Exhibiting a significant delay in flowering time. Here, the most effective line is the MSFTA1074, which was shown to flower 21 days later than the empty vector control under our long day extended conditions. Here, this extended vegetative growth period was directly translated into having a substantial increase in biomass. Continuing on, I have another data set from the primary research article that shows the mutant lines having up to a 78% increase in fresh weight and a 76% increase in dry biomass yield with no associated weakness in either plant health or structure. Additionally, the forage quality of the mutant lines were also enhanced as our biochemical analysis revealed a significant reduction in stem lignin content and acid detergent fiber. With this, the mutant plants also maintained a higher level of crude protein and showed improved in vitro dry matter digestibility. This combination of traits indicates that this genetically modified alfalfa was not only more abundant, but was also more nutritious and digestible for livestock. Furthermore, this study is significant because it demonstrates a successful translation of CRISPR technology from model systems to a complex and commercially important crop. Here, there's a dual improvement in yield and quality, with the multiplex gRNA strategy effectively overcoming the challenge of polyploidy. With this, further work should focus on validating these results across different genetic backgrounds and environments. And additionally, there should also be some focus on assessing the potential ecological impacts of this, such as effects on local pollinator populations, as the plants won't be flowering as often, so there might be less pollinators. Furthermore, these late flowering lines also present an ideal genetic background for stacking additional traits. One such example of this would be stacking abiotic stress tolerance on top, and then having alfalfa plants with a multitude of genetic enhancements. In summary, 3 main conclusions can be drawn from this article. First, that the FT Florigen system serves as a conserved and potent leverage point for manipulating plant development. Secondly, CRISPR-Cas9 provides the precision to engineer this pathway effectively, which surpasses earlier transgenic methods. Lastly, as evidenced by the works of Rulabu et al., targeted gene editing can directly address the yield quality trade-offs, which offers a powerful strategy for crop improvement. And with this, I have my list of references where I obtained my information, my images, and my primary research article. Thank you.